otwiera, bo ja skupiałam, żeby dołączyć do celu. Chyba wszystko jest okej. Już nie będzie aż takiego presji czasu. Good morning. Can I have your attention, please? Good morning. Please take a seat. We are about to begin, so find your seat and we can start. Welcome to the Institute of Bioorganic Chemistry, Polish Academy of Sciences in Poznań. Thank you all for being here today. My name is Małgorzata Marcinkowska-Swojak, and I will have a pleasure to host the sessions today. And without further ado, let me introduce the director of the Institute and the head of our Genomics for Poland project, and she will share a few opening remarks. So, ladies and gentlemen, Professor Luisa Hanszu.
Thank you very much, Gosia. I'm really happy that we are here all together in this great day. In fact, I believe that great things start on usual days. So this day is usual, but I think in future we will see it was not so usual. So I hope we will do together great things. And first, at the beginning, I would like to warmly welcome all our prominent guests. And I would like to start from our prominent guest from abroad. So Professor Andre Uterlinden, the head of the Laboratory of the Population Genomics, Erasmus University Rotterdam Medical Center, Netherlands, also the leader of the Genome of Europe project and our first prelegent today. Andre, you are welcome. I'm also very glad that Professor Ivo Gut, the director of the National Center for Genomic Analysis in Barcelona, Spain, is with us today as a guest. He is also engaged in the Genome of Europe project, work package leader, and I found that a few days ago he was elected a fellow of the European Academy of Cancer Sciences, one of Europe's most prestigious scientific institutions dedicated to cancer research. So we are very happy you are here with us, Ivo. Then I would like to warmly welcome the distinguished guests from Poland. First of all, our second prelegent today, Professor Jan Lubiński from the Department of Genetics and Pathomorphology, Faculty of Pharmacy, Medical Biotechnology and Laboratory Medicine from the Pomeranian Medical University in Szczecin. We are welcome, Professor. I would like also to welcome Professor Jerzy Duszyński, the former president of the Polish Academy of Sciences and former director of Nęcki Institute. I welcome Professor Andrzej Legocki, former president of the Polish Academy of Sciences and former director of the Institute of Bioorganic Chemistry. And I hope Professor Jerzy Kaczorowski is also with us. I don't see, he promised to be, maybe he will join later. He's the president of the Poznań branch of the Polish Academy of Sciences. So we have quite good representation of the Polish Academy of Sciences here. I would like also to welcome Professor Marek Figlerowicz, the former director of the Institute of Bioorganic Chemistry, Polish Academy of Sciences, head of the Department of Molecular and Systems Biology, who was a pioneer of genomics in our institute. I would also like to welcome Professor Marzena Cichomska and Professor Magdalena Duch-Wiszerek from the National Institute of Geriatrics, Rheumatology and Rehabilitation in Warsaw. I also welcome Professor Łukasz Łaczmański, the Deputy Director for Scientific Affairs of the Ludwig Hirschfeld Institute of Immunology and Experimental Therapy of the Polish Academy of Sciences in Wrocław. And then I would like to welcome our hematologists, which collaborate with us for a long time. Professor Krzysztof Lewandowski, the head of the Department of Hematology and Bone Marrow Transplantation, Poznań University of Medical Sciences. And also hematologist Professor Jacek Wachowiak. I would like to also welcome members of the Scientific Council of our institutes and, of course, consortium partners, so Genomics for Poland consortium partners. And please forgive me, I will welcome only one person per one team, but of course I will welcome you, all of you, cordially. So from the Institute of Human Genetics, we have a team with Professor Natalia Rozwadowska, the Deputy Director for Development of the Institute. From Lęcki Institute of Experimental Biology, we have the director, Professor Agnieszka Dobrzyń. Poznań University of Technology is represented, I think, by Professor Aleksandra Świecz and Dr. Paweł Wojciechowski. University of Warsaw, a Center of New Technologies, is represented by Dr. Dorota Adamska. Medical University of Białystok with Dr. Agnieszka Ostrowska. University of Łódź, Dr. Błażej Marciniak. And from University of Gdańsk, Professor Grzegorz Węgrzyn. I don't see, okay. But hopefully he will come. And from the Jagiellonian University in Krakow, Professor Jolanta Jura, the Dean of the Faculty of Biochemistry, Biophysics and Biotechnology. And we also have representatives of clinical units, so Cardinal Stefan Wyszyński National Institute of Cardiology, National Research Institute in Warsaw, and the representative is Professor Tomasz Hryniewiecki, former director of the institute. And from Maria Skłodowska-Curie National Research Institute of Oncology, Gliwice branch, Professor Małgorzata Oczko-Wojciechowska, head of the Department of the Genetic Diagnostics. And we also have in our consortium one partner from Łukasiewicz Research Network, Polish Center for Technology Development, and I hope that Dr. Paweł Sega is with us. Okay. I invited to this meeting many local and government authorities, but unfortunately they decided not to come, except for one or two persons. So I hope Mrs. Milena Wawrzynowicz is with us here. No, she's the director of the health department of the Marshal's office in Poznań, and she declared to be here with us. Also is Magdalena Rozumek, who is the representative of the same department. And Natalia Ostenzaken is here. I don't see. She's representing the member of the Republic of Poland Parliament, Jacek Tomczak. I would like also to welcome the representative of Analytic Genetica, Juliusz Unruk. So this is our business partner in the project. And I would also like to welcome all of employees, doctoral students of our institutes, also retired professors for example, Professor Michał Sikorski. I'm really glad you are here with us, and I hope it will be a great day also for you. So all other guests and friends who decided to come here today are also warmly welcomed. European Commission regrets but was not able to participate in the kickoff meeting of our project. They, however, sent me a short sentence, which I decided to read here. So the European Commission supports the implementation of the Genomics for Poland project, financed under Cohesion Policy, addressing the need for a coherent and interoperable infrastructure for genomic data. The project contributes to advances in personalized medicine and innovative therapies, while reinforcing Poland's position with the European research ecosystem and delivering tangible benefits for citizens. So I think this is the end part of our first official part of the meeting, and I think we can start our lectures today. Thank you very much.
Thank you very much, Professor Hanszu, for this warm welcome. Now we are moving to our presentations of our guests. So I'm delighted to invite Professor Andrea Batalinden, who is one of the world's most influential scientists in the field of complex genetics. As director of Genomics facility at Erasmus MC and the coordinator of the very ambitious Genome of Europe project, he is the forefront of establishing a massive genome reference database for the whole continent. He is a primarily architect of work related with population studies, including the Rotterdam study and Generation R, and his pioneering work continues to bridge the gap between large-scale population genomics and the practical application of precision medicine in global health. So we are eager to hear your insights. Professor, the stage is yours.
Where can I? This one. Oh, that's yours, yeah. Okay, there we go. Yeah, okay. Well, thank you very much for this invitation. I'm deeply honored to be here. And first of all, I would like to congratulate you with the fantastic project you've been able to generate. So big applause for Louisa. I think it's very important for you also. Of course, we like very much to participate in Genome of Europe, but I think it's also very important for Poland to have this technology and knowledge being implemented and to collaborate on the European level, especially in this day and age, as we all realize here. I will take you today through a little journey of explaining why we are doing all this kind of work of sequencing DNA and implementing genomics in healthcare. It's done by science, and this shows you how science actually works. You see here on the right that we search, but sometimes with imperfect tools, and then we find something, and you see this illustrated by the disc of Festos, which is still not translated. It has been found quite a while ago, but because we do not have a reference to compare it with, we cannot translate it because it's a single language we see, as opposed to the stone of Rosetta. Because that we have 3 languages, so we have references to compare, and that's how we understand those languages and how the Egyptian hieroglyphs was deciphered. So that's why we very much need reference data in order to be able to interpret and understand all the data. And the same thing holds for DNA, of course. Then we start to implement it, so we found fantastic discoveries and we want to use them in practice. We want to apply it in healthcare and prevention maybe. But then, of course, there are always 2 sides to it. Of course, Hippocrates. We want to do the best for the patients and people in general, but there might also be some dark sides to it. Is it really beneficial for the population to use this technology on a large scale, perhaps? So that's why we still need discussions also with people from other disciplines, sociology, ethics, philosophy, to help us with interpreting how can we best use this kind of knowledge and discoveries. Well, as you see, I'm coordinating the Genome of Europe, so that will be a major part of my talk. Before, I would like to show you my disclosures. Usually people flash this slide away, but I'd like to explain it to you a little bit more. I do not hold stock in any company, and I receive only 1 salary from the Erasmus Medical Center, and I just retired also. I'm emeritus since 1 month. I get my funding for research from public sources, and sometimes companies pay me to give advice and attend meetings and give lectures like this. So these are my disclosures to you. This is our starting point. Look at your neighbor. Yeah, we're all different. And you see here some fine examples, some of whom you will recognize and some of whom might be less known to you. And that's, of course, the curiosity we have to understand. What are these differences? Why are they there? And what do they mean? Of course, there are many types of differences. You see here some people which differ very much in behavior, if you look at the right low corner. But of course, I'm working at a medical center, like most of you will be doing. So we're very interested in why do some people and how do some people develop type 2 diabetes rather than prostate cancer or Alzheimer or the other way around. And that's, of course, very important and informative for us to design treatments and diagnostics in order to prevent and to help those people. Now, to summarize a lot of research being done over the past 50, 60 decades is that we learned that variation in the DNA molecule, and I don't have to explain this to you, is very fundamental to this. It contributes to these differences. It doesn't explain it fully, but it contributes to these differences. And that holds for the very different, let's say, phenomena we see in humans, but also in other species, of course, like the development. Babies, when they're born, they're already different, and the way they develop, of course. Appearance is driven by DNA variation. You might notice, why is that important? Well, for example, for forensic, that's taking a very high-speed development right now. Because forensic scientists are using DNA traces to understand if this perpetrator had blue eyes, attached earlobes, and dark hair. And that can help, of course, the prosecution of those criminals. And we've seen some fantastic examples of that. Also through looking at DNA relationships in the DNA database, but we can discuss during coffee break. I explained to you already behavior, so some fields have really been improved in terms of their scientific level by starting to do genetic studies, because they require quite tough rigor and collaboration also, which was not the case, for example, in the field of psychology, where you had a lot of isolated small studies, and now through genetics they started to work in large-scale consortia, harmonize data, etc., and that has really improved the science. And then last but certainly not least, aging, and which is sort of a dustbin for all the diseases we especially experience at old age. We now understand that many, if not all of those diseases are under genetic control, meaning again, genetic variation plays a role, contributes to the differences in appearance of those diseases. Well, all those developments have led to this concept of what is called precision or personalized medicine. Mostly when I speak to clinicians, they say, we've been doing personalized medicine already for 2,000 years, so that's not new. We treat patients, we don't treat groups of patients. So precision medicine might be the better term to use in this respect. So again, why do we study DNA variation? Why not simply look at blood markers or phenotype variations? And these arguments, they hold both for very rare DNA variations as well as for very common DNA variations. They help us to understand biology, DNA, blueprint of life, that determines how proteins are encoded, how they work, how they function, in which cells they are expressed, under which conditions. So that will lead you to understand mechanisms. And if you understand mechanisms, you can intervene in those mechanisms and you can design treatments and interventions. Not necessarily drugs developed by pharmaceutical companies. Also lifestyle interventions can be driven by understanding the mechanism of this through DNA studies. On the other hand, you have, of course, the powerful application of prediction. Once you know that a certain DNA variation or a set of DNA variations is related to a certain outcome, let's say a particular disease, you can determine that DNA variation at a certain point in somebody's life and use it to predict. Now, there are big debates about it. When and how should we do it? I remember last evening, during a fantastic dinner, we had a discussion on that already. And that's very important to have that debate among each other, not only with the specialist, the clinical specialist, but again also with other disciplines to help us inform what is best for the population and for the individual patient to do that. But of course, you can imagine in a clinical setting, this offers fantastic advantages to do a very early, appropriate diagnosis. And the big advantage, of course, is that DNA is a very stable marker. So that relationship is very stable throughout life. That helps you to predict risk of disease, and that's where most of our findings actually come from now. But it also would be very helpful to predict progression. So once you have a patient, you want to know, is this a fast progressor or a slow progressor? And of course, DNA information can be very helpful for that, but there's not that many DNA variants available at the moment. And last and certainly not least is pharmacogenetics. So we can predict response to treatment for certain drug gene combinations. And that has been a field that has been always on the front of applying this in clinical practice and maybe in an even wider setting in the population. But still it has not reached maturity. Due to various reasons, again, I won't go into that here, but it's quite puzzling why it is not applied yet very massively in all kinds of countries. And we're having big debates about that also. So this tells you why we should do this study. It's very important. Always ask, especially for the young people, why am I doing this? Don't forget that. Another big starting point is, of course, how much DNA variation is there? Is there enough in order to do such studies and to make discoveries? Well, answer is yes, no doubt. There was a very nice review in nucleic acid research last year, where they summarized all the progress made in erecting and filling up the DB-SNP database. And as you can see on the left corner, there were several projects that contributed to that by sequencing human genomes, like we're doing now in the Genome of Europe. And of course, we had very well-known projects, the HapMap project, of course, the CEF before this in France, 1,000 Genomes, Exact, Genomed, etc. And they all contributed to understanding how much DNA variation there is among human individuals. On the right graph, you see what the volume is of that. If we do the counting now, we have over 1 billion nucleotides in the 3.3 billion nucleotide DNA molecule that can vary between people. Yeah? So that's 1/3 of our DNA sequence can vary between human individuals. That's enormous. Most of them are very rare, of course, but the volume is enormous. And this is based only on about 6 million genomes that we have sequenced in history now. 6 million out of 7 billion people. So we've only scratched the surface of DNA variation. There's an enormous amount of variation still to be uncovered. And that's, of course, very hopeful for especially projects like Genome of Europe. We've also learned that there are a few techniques that allow you to uncover what kind of relationship there is between the DNA variation and a certain outcome. And that is, in fact, a function of the frequency of that variant and the effect size of that particular variant. If you go to the left upper corner, you will see the Mendelian diseases. So these are the classical workhorse of clinical genetics. You have a pedigree where a phenotype, a disease is segregating in, and usually you will find after sequencing, and especially now in a few weeks or a few days maybe even, the causative mutation for that. So that's a very rare DNA variant with a very big effect, and that allows you to follow it, segregating according to the Mendelian laws. And now we have several techniques that allow us to find those variations, those mutations, very efficiently and rapidly. There are about 8,000 of these rare diseases. I would say we have uncovered the cause of that for more than half, maybe 2/3 of them already. There's still some unknowns of there, but the question is, are those real Mendelian diseases? But if you move to the other side of the spectrum, so to the more common variations, we have noted by now, because we've done massive studies on that the effect sizes of individual variants there is much, much smaller, which makes, of course, a lot of sense from an evolutionary perspective, because very detrimental variants cannot be tolerated in a population. But their small effect size were initially ignored because we look at them individually and said, okay, what's an odds ratio of 1.01? That's insignificant, ignore them. But now we know that for certain diseases, there are not 1 or 2 variants available there, but hundreds, if not thousands of them. And if you combine them into what is now called polygenic risk scores, the effect size of those polygenic risk scores, if you're on the top of the distribution, is equal to that of Mendelian mutations. So now the clinical geneticists also became aware, this is important, we should start working with that. Those were uncovered by what we call genome-wide association studies, and as was mentioned already, I've been a part of those discoveries through the contribution of the biobanks and the cohort studies we have in our case at the Erasmus Medical Center. And this is how we also set up international collaborations to work together because of these very small effect sizes. We need to combine loads of data in order to find those effects and then later on establish those polygenic risk scores. So we now know that the Mendelian diseases can very quickly, through sequencing, deliver actionable variants, and actionable meaning you can use them for diagnosis. And sometimes, and this is a big pitfall, of course, of Mendelian diseases, they are available for treatment, for example, through gene therapy. But those are the exceptions rather than the rules. For most of the Mendelian diseases, there is no treatment available yet. And that's very different from the complex diseases, because these are typically gene-environment interactions. So there's a genetic predisposition, but on top of that, there's environmental factors that influence the expression. And those can be the diet you take, the exercise you take, the lifestyle you have, etc., etc. And those things, of course, are amenable to change. Yeah? You can advise people to change their diet. Now, we also know human psychology. It's not easy to ask people to change their diet unless they have a very strong reason to do that. And we think maybe genetic information might be helpful in there. Because, that's inborn, you cannot change your genetics. And that's a big risk you have with you, and a big warning maybe. So maybe this will help to change lifestyles in any way. As I said, these are now, let's say, led in their application through the development of these polygenic risk scores, PRSs, and those are the big buzzwords we have now in complex genetics. So two approaches for rare and common variants, and of course they overlap, and eventually they lead what we call the genetic architecture of a disease or a phenotype. And that's illustrated here. It's a convoluted slide, I apologize for that, but what you see here is the distribution of the values of that phenotype, let's say height or cholesterol level, maybe easier. So you have very few people with very extreme levels, either very low or very high, and of course the bunch of the population will have a normal level in between. And that holds not only for risk factors like cholesterol, but also for diseases like cardiovascular disease, osteoporosis, what have you. And if we then look at the genetics, we now know that in the extremes, those are explained by Mendelian mutations, because those are very strong effects, are very rare, and lead to very extreme phenotypes. But if you move through the normal population distribution, you will see that most of them are explained by these polygenic risk scores. So these sets of common variants that have little effects, but combine, explain a lot of the variation in those. And we've developed polygenic risk scores for many of those phenotypes and diseases by now, and together they form, let's say, the architecture of a disease. So a disease is never one single mutation that explains it from a genetic perspective, but it's always a combination of rare and common variants together explaining the variation in the population. And that also leads to a new view on genetics, which is much more holistic, so to speak. So you're not looking at a single mutation anymore, but we're looking at the genome, and there's a strong mutation in there that has a big effect, but that is helped by the rest of the genome. They influence the expression of that one single strong mutation. And that is also used now in clinical practice in several centers across the world, where people not only look at the Mendelian mutation, but also in context of these polygenic risk scores in how they modify the expression of those Mendelian mutations. Well, very important in all of these fantastic discoveries, I'm sure you agree, is technology. Technology always has been a very pushing force in our field. Through the development of new technology, suddenly many more discoveries were made. I will talk about sequencing, of course, but I also like to mention arrays, which is sometimes paid less attention to, but they played a very instrumental role, especially in the GWAS activities. As you can see here, that's illustrated simply by the price of doing an assay. So initially in 2005, 6, 7, the price of doing one sample with an array was about €1,000. So those were very costly experiments, and that's why there was a lot of critique at that moment of why are we spending so much money on finding these SNPs with these very tiny effects? Why is it important? So I and several colleagues had to defend that approach, and luckily we were right, and then we showed and we delivered. But it was also driven by this cost. And as you can see, during time, and that has to do with people like me and Ivo talking to those companies saying, fantastic technology, but your price should get down because that's good for you, because you can sell more, and it's good for us because we get more data. And in the end, that will be good for humanity because we make more discoveries. So in the end, in this case for arrays, they listened in 2016, and they made us an offer we couldn't refuse. In this case, Illumina, but also Affymetrix, they said, okay, if you buy 500,000 arrays, we'll give you a good price, lower than €30. I said, oh my God, 500,000 arrays, how do I do that? But by that time, I had established quite a big network of collaborators, and so I reached out to them and say, look, if you have your samples genotyped, in this case by 3 centers in Europe, in Rotterdam, Paris, and in Bonn, we can offer you this price for that sample. And the success was enormous because I got over 1 million samples to be genotyped within 3 months. So that was a big success, and also showing how important is the ecosystem of talking to those industrial partners and making things work for both parties. By now, a few words on the arrays. I'm a big array fan because I think they're very robust technology, which allow us to do the first implementation of genetic information in healthcare and prevention, because they're very much cheaper than sequencing at the moment. Of course, they're approaching each other, but they will stay very cheap. We're currently talking about prices of less than 10 euros for doing an array analysis. You can customize them so that you can totally fill them with variants you would like to see. And of course, the ones that we would like to see are the ones that are actionable. So once we see them, we know what to do with that variant, rather than have what is called a variant of unknown significance, a VUS, which are the nightmare of all clinical geneticists, of course. The technology is very robust, high throughput, data analysis is very straightforward, low IT footprint, so you don't have to spend loads of money on your servers for storing the data and handling the data. And that leads to a lot of applications in clinical practice. I'll show you one example. We're following up in Erasmus called the GOAL project, genotyping on all patients through arrays at the moment. But of course, we're keeping track of what the technologies are doing. Maybe now the arrays are popular for this, but maybe that might change by sequencing or maybe other technology coming around. So that again illustrates why technology and industrial partners are very important in this era. A quick slide on one of the critiques we got for applying arrays was you cannot use them for genotyping rare variants. And we and others show through many studies, some of which are published, and now you see a list there for different applications, that's nonsense. You can genotype very rare variants very accurately with arrays. Of course, they require optimization, but it can be done, and still for this very low price in there. And that's why there's now a revived interest, for example, in reviving the screening for familial hypercholesterolemia, because that's a very easy to detect, let's say, disease. Only 3 genes, many mutations we know. There's a very cheap treatment available, statins. So that's a no-brainer, I would say. We had screening programs in the Netherlands, but they were stopped because of the high cost of sequencing, which was used for that. But now people are returning to arrays because it's much cheaper. We can fill it with all the mutations we would like to see. And now we have pilot experiments running in the Netherlands, but also in other countries on this. And you see some other examples also where arrays are used. For example, in pharmacogenetics, also for a very low price. So that leads to, let's say, an example of technology which can be used in clinical practice, which is undergoing evaluation by authorities also, how valid is it and how good is it, what kind of controls do we need, etc. And of course, and I'm Dutch, it's very cost efficient. Yeah? You don't have to pay too much and get a lot of information which is useful for that. Yeah. Okay, and back to progress in science. This illustrates until 22, I update this every 6 years. To see how the progress is in GWAS, finding those variants involved in complex diseases like type 2 diabetes, breast cancer, etc. If you see on the left, 2010, when the first big GWASs were done through these large international consortia, thousands, 100,000s of people were involved. We found a few dozen of hits, but they explained a modest amount of variation, genetic variation of these diseases, so they were not applicable. But we did notice that by increasing the sample size, we could find more of those variants. So that happened in those following years, and you can see the progress in there. Take, for example, height, the easiest example. The biggest consortium we have at the moment is the GIANT consortium working on the genetics of height. And as you can see here, that was published 2 years ago now in Nature. Genotyping more than 5 million people led us to understand the genetic architecture of height. We almost completely understand that now, what the genetic basis is of height. I'm a tall Dutch person, so I have a personal interest in what is the genetics of height, so that's why I'm involved in those studies also. But it's an example. It's not directly clinically actionable, of course, height. It's not used in the clinic. However, in one of those GWASs, we did do a finding which has led to a new treatment for small stature at birth. So telling you that those GWASs are not only used for finding small effects, but also lead to new treatments for which are clinically very useful for that. Well, that story also holds true for all these other diseases and traits. Simply, we have not reached those numbers yet because of lack of collaboration or lack of sample collections, etc. So that's why it's so important to have those biobanks available in your country where you collect those samples, well phenotyped, well documented, so that at some point you can be able to contribute to these large efforts. As you can see here for breast cancer, we have quite good prediction now with the 313 PRS. Macular degeneration always been a post-stamp example for that, which is also used in clinical settings right now to predict risk of macular degeneration and offer lifestyle changes in there based on that. So how can we start applying polygenic risk scores? Well, I told you in earlier slides, you can use it for a more holistic view on the genome when you have a patient with a genetic disease. So you see those mutations, but also take into account the rest of the genome through those polygenic risk scores. But I think maybe an even more important application will be the prevention, so the public health. And that's illustrated here. You see on the top in the red boxes the most prominent diseases where we have very good PRSs for at the moment, based on a paper by Nailen in Nature Medicine not so long ago. And you see the dots, which sort of reflect the distribution you will have once you do a group of people, people, not patients, but people, and genotype them for a certain PRS. You get a distribution like that. You have on the left, you have people in the low risk, so they have a very low genetic risk for this particular disease. In the middle, you have the people who have a normal risk, the population-based risk for that. And on the right, you see the people who have a genetically determined very high risk for that disease, driven by this PRS. So what can we do? Well, as I explained to you already, if you're in the top 1% or 5%, depending on the PRS, of that distribution, that effect is equal to a Mendelian mutation. So that's clinically very relevant. It's a very strong effect. So if you translated them back to prevention measures, what can you do? I'm not sure. Is there a mammography screening program here in Poland for breast cancer? Yes, you have? Okay. So the idea now is that we have this mammography. Usually it's done for women at age 50. You call them to undergo mammography every 2 years until they reach 70 or 75, because then it doesn't have any added value. The idea now is to first do a genetic test, stratify those women in those 3 categories, low, middle, and high risk, and then base the mammography screening on that. So the low-risk women will not undergo mammography because it doesn't have any added value for them. So you will save them the burden of undergoing mammography, which is not pleasant, of course. The middle group will undergo the normal screening modality, 50 years and older every 2 years, but the high-risk women will immediately enter into the mammography screening. Because in our case in the Netherlands, if you do the normal mammography screening program, you miss 20% of breast cancer still, 20%, 1 in 5. That means we have a lot of women which have tumors arising much earlier. And we hope that such a genetic test at a much younger age can pick those women up to a large extent through the combination of genetic testing and mammography. Now, this is an example for existing screening programs for breast cancer. And personally, I'm a biased person, of course. I think the beauty of it is once you do such a genetic test, it's all on the DNA. This single DNA test can also be used to extract information for colon cancer risk, for familial hypercholesterolemia, for your pharmacogenetic profile, all from the same single genetic test, if you do it with arrays very cheaply, and then use that information to steer people into particular screening programs or give them help, like with a pharmacogenetic profile, to undergo the best treatment based on that pharmacogenetic information. So that's a proposal that is now undergoing debate, of course, because it's quite a shift in the whole healthcare system, which, as you know, is very siloed. You either have diabetes or cancer, but both at the same time is a problem for most healthcare systems in reimbursement, in treatment, etc. So we hope to start making let's say, a rupture in that by introducing this single test, which allows you to predict many different kinds of diseases. I hope we'll have many more such discussions within society also about applying this, because I think it can be of great value for individual patients and people if we do this in prevention programs. I told you we have a program in a clinical center where we are using this type of information in clinical settings. In this case, at the Erasmus Medical Center. This is not happening, by the way, at the moment. We're only doing pilot projects. But the idea is that every patient that goes into the hospital first gets a genetic test, so genome first. That genetic test then extracts information which is distributed to the acting clinician to inform about the diagnosis, about any other findings which are clinically actionable. For example, you come in with a broken leg, but you do see through the genetic test that you're an FH carrier, so we can offer statin treatment to prevent cardiovascular events later on. That clinician discusses those findings with the patient, is helped with the diagnosis, the treatment through the pharmacogenetic information, and also the patient gets some lifestyle advice on the risk for type 2 diabetes, for example. This is the ideal setting. So we're exploring this to implement this. And that's a big thing, because we can make fantastic discoveries, everybody enthusiastic, but to actually make it implementing in an existing healthcare system is a big challenge. It's way beyond my pay grade and knowledge, so I leave it to other people to work on. But it comes with many different challenges and problems. But still, it's driven by this knowledge, this can help people. Yeah, it can help patients, it can help healthy people to not get sick. So this is an idea we are piloting at the moment with several different clinical departments. You see them listed here for eye disease, breast cancer, cardiovascular disease, the pharmacogenetics, of course, and locomotor diseases. We are doing that with a genomics core facility we've established in the Erasmus Medical Center, where all the knowledge and expertise and the equipment is centralized, much like you're doing here. And that will help, of course, to get all the people together from different disciplines working on this problem. Also under ISO certification from the departments, not so much from the core facility here. We have some ongoing projects already, like we're doing epigenetic profiling of tumors, which are very important to inform the surgeon about operation and removing the tumor. We're doing epigenetic profiling for clinical genetics, and of course we do CFV typing for finding Mendelian deviations. The core facility is also exploring now the diagnostic use of polygenic risk scores in close collaboration with the Department of Clinical Genetics, for breast cancer, but also with the Department of Ophthalmology for macular degeneration, because they have very strong PRSs for glaucoma, for macular degeneration, and that's a big opportunity there because there are also treatments attached to that, so that's very promising. And of course, we have ongoing trials now. I was explaining you the example of breast cancer that's being trialed now in several studies around the globe, and the first results are coming out showing that it's not worse than existing programs, but we have ongoing trials now to hopefully show that it will also improve the outcome if you look at long-term survival of those women. So very promising developments in there. Well, this sort of summarizes what we're currently thinking of applying genetic information, either by sequencing or by arrays or by other techniques, but genetic information, which you can determine at any point in life. So we can discuss about doing this at childbirth, heel prick, do genetic studies on that. Is that a good idea, yes or no? Big debates. Should we do it with all people when they're 20 or 30 and inform them about the risk for cancers or for diseases we can treat actually at the moment? Should we do that also for diseases which are not treatable at the moment, like Alzheimer's disease? It's always a debate. Usually when I ask students, there are always one or two who say, yes, I want to know my Alzheimer's risk, whereas you would expect nobody would want to know that, but still they do. And of course you have those screening programs I told you about, especially for cancer. I think there are big opportunities there. I will not go into the applications outside of clinical science because that's a special field, forensics there. So there are many potential applications of this information. And that brings us back to Genome of Europe, because we were looking at this situation quite a while ago and asking ourselves, how much genetic data is there to do all these studies? So do we have enough? That was the simple question. So first question is, how much data is there? That's shown here. If you look in history, there's not so much data. Especially not for sequencing, you showed here. There are about 6 million human genomes sequenced at the moment, available in databases. The situation is better for arrays because there are over 100 million people who have had their genome genotyped. The bad news is that it's in the hand of commercial companies for about half of these samples. And even worse news now is that it's based in US companies, which are not our friends, colleagues, maybe anymore. So, although there's lots of data and we collaborate, we do, but there's not that much data actually. If you look at what kind of projects are generating those data, especially the UK and USA are leading those efforts. UK has Genomics England, UK Biobank, Our Future Health now with 5 million people. USA had programs like TopMed, All of Us program, Millions Veterans program. And then you have others, and you see Iceland, for example, a shining example in Europe and some other countries around the world. But you will notice Europe is lagging behind. Europe has a lack of countries working on this until now. The other question, of course, important to answer is how much do we need actually? Do we need billions of people to be sequenced or genotyped? Well, that's also driven by this knowledge because we know now, I showed you this progress in Jiwa's slide, that for arrays, we indeed need millions of samples to be genotyped to understand all of the genetic variation in there. For height, I showed you that, 5 million people. Based on that, we did some calculations with people who are experts in that, and that leads to guesstimates that, for example, if you want to do this for type 2 diabetes, you need about 50 million people to understand the complete genetic architecture of type 2 diabetes. And that figure goes even higher for even more complex diseases with much more environmental influences there. So indeed, we need millions more of samples with good phenotype, but also with good genotype to enter that. That is based on reference data. Again, reference data are very important. So that same calculation led us to understand we need many more reference genomes, especially if we want to apply this in Europe, because most of the data is coming from UK and USA, and that's not reflecting the European genetic diversity. So yes, we need many more samples, as shown here, and that runs into the dozens of millions of samples with good genotype and good phenotype there. So we did a first scan of, how much data is there around in Europe, and we did it together with Nicole Soranzo from Italy, Milan, and Taras Oleksik, a Ukrainian scientist, also working on this. And that led to this map of Europe, where you see the density of genetic data per capita. And so Iceland, you see in yellow, so per capita there's an enormous amount of genetic data there. Few inhabitants, lots of genetic data. But you see if you go south and to the east of Europe, it's a much less optimistic situation. So there's a lot to be done there. So the information, genetic information available is very low in Europe, and that leads to the very simple conclusion. We need to sequence and genotype European samples in Europe. Yeah. We were very fortunate that same conclusion actually was drawn by Mario Draghi from a totally different field. He's a banker, you might know him. He saved the euro in 2008, very important person. He wrote a very influential report in Brussels now about the competitiveness of Europe. And that report is the Bible in Brussels at the moment, I can tell you. They follow that report quite literally. As Trump does with Plan 2025, Brussels is following the Draghi report. And that report mentioned the 1 million genomes as an example where we can collect and generate such data in. So that has led to, among others, to this 1 million genomes initiative, which started in fact earlier in 2018. It's a little bit out of member states. It goes through states, countries, with ministries signing declarations. Yes, we want to make 1 million genomes accessible and available for research and application in healthcare. We have a strategic overview group, the coordination group, so myself and Ivo are part of that, where we advise on it. That's been split into expert working groups, and that's where the details are being discussed, like the technological developments, what kind of IT infrastructure do we need in order to store such data, to make it available. And we have defined a few use cases where this information can be almost immediately applied, like for rare diseases, like for cancer, but also for complex diseases, as I showed you. So this last working group 12 is the Genome of Europe, and that was a spin-out out of the complex disease working group number 10. And now we have started a new working group, the pharmacogenetics working group, which is taking the place of Genome of Europe. So the working group 12 now is pharmacogenetics, but it used to be Genome of Europe. You see here the map of Europe, and all of the countries are involved, except for a few observer countries. And Poland is still an observer country, but I hope to work on that to make you a real partner in that, and we'll make it happen. But also UK and Switzerland are observer countries, and UK I do not have to discuss here, but also in Switzerland there are many activities to contribute to Genome of Europe there, almost out. Oh, there we go, yeah. So what happened in the 1 million genomes is we have a few big projects that's based on the IT, setting up this IT infrastructure that's called the European Genomic Data Infrastructure, the GDI project. Budget of 40 million, a lot of experts on IT stuff talking to each other how to make those data, to store it and to make it accessible. The other project is the Genome of Europe, also with a substantial budget to create data. The current budget allows us to generate around 100,000 genomes across all those countries, but I'll explain to you in a few moments. Below and on top of that, we have coordination efforts where, again, those issues that I showed you in the opening slide are still being discussed. What kind of use cases can we think of, how to coordinate the IT setup, which is now reaching maturity, how to store those Genome of Europe data, etc. But very important in all those efforts is to realize that we are supported by taxpayer money. So this is your money we're burning, so we do this very responsibly, of course. And we always have to report on how we spend the money, for what purpose, and for what activities we do with that. So it's very important to realize that this is being paid by the citizens of Europe. It's also a matching scheme, I forgot to mention here, so that means we are in the Digital Europe Directorate, and that is a 50% matching funding scheme, so you make 100% cost and only half of that is being refunded by the European Union, meaning that the other half have to be funded by you. Although there's disadvantages for research, but it promotes activity within individual countries, like is happening here now. And we've seen that to work out quite well, in fact, across Europe. Okay, a few words about the layout of the Genome of Europe. So we want to make a representative collection of European genomes per country. So you want to establish national reference genome collections for each of the countries, but in the end combine them. We want to do it for subpopulation in each country. For example, in the Netherlands, we have a rich colonial history, so we have white Caucasian Dutch people living there, but of course, also many colonial people from Indonesia, from Suriname, all those other places we conquered centuries ago. They're living in those countries, so they are healthcare consumers, so we also should generate reference genomes for them. We also are working with countries which are outside of Europe, the European Union, but which are living in European countries, like Turkish people, for example. Not an EU member, but many Turkish people live in many different European countries. So we should also generate Turkish reference genomes. And the same holds for those which are not represented by a country, like the Roma people, migrating people, or the Sami people in the Scandinavian countries, travelers in Ireland. Very important to take those also into account. There are usually special groups with their own culture. So we're setting up a minority board to involve them. So not over their heads do this, but with them and make sure they profit from these developments. I will show you in one slide later about the use cases we have developed, but the main focus of the project really is on generating those data for reference purposes, and that's it. So a very simple project, in fact. We had a great kickoff meeting in Rotterdam on a boat, very symbolic, of course, because we are undertaking a journey together. So here you see the brightest minds in Europe on genetics and genomics together, working on this project, spending their energy on it. So it was a fantastic meeting, and I think you're here. Lisa? So you were there. It was fantastic to have you all together because it's really important to collaborate with each other, to understand each other, the challenges, but also the commonalities we have among each other. Well, it's a straightforward project with work packages. I won't go into detail there, but of course on the different aspects, how to collect the samples, the biobanking, how to sequence the samples, and Ivo is leading the technology work package on what is the best technology and how to harmonize it. That's very important to do that. And that will lead to a fantastic infrastructure in Europe where we have collaborating sequencing laboratories, which are doing more or less the same thing and can help each other with that. You see here on the top, our funding allows us to do about 100,000 genomes, but I've received good news from several contributing countries, including Poland, so that number will probably be higher. And it's coming from different sources, I'll show in a moment. We are technology agnostic, so we're not focused on one technology, one sequencing center. That would be easy, but we've deliberately chosen to involve all kinds of sequencing centers and to adapt to the different technologies which are available. We really want to focus on general citizens as a normal population, so not patient-based collections, but general citizens, and it should be proportional. So we will sequence more German samples than samples from Malta, for example. And we will focus on ancestral backgrounds, well, I'll show you that in a moment. This is the layout of the numbers, because we said we want to make it proportional, so we looked at how many inhabitants do those countries have. You see them listed here. I think Poland is in there for 38 million, I don't know if that's correct. And if you translate that then back to 500,000 samples, because that was the original plan, so the budget now is only for 100,000, but the original plan was for 500,000. You see that translates to a number of genomes we are expecting the countries to deliver to make it proportional and representative in this Genome of Europe collection. So the Netherlands is in there for 22,000 genomes. That should be our original focus on that. But the money only allows it now to do about 5,000 genomes. But that's about the layout we have designed. On the left, you see the type of origin of the samples we can accept. Legacy data, so existing data, might be amenable, but we have to check, and we have the genome police sitting there to check, is this good sequence data? Is it allowed to enter the database? Yes, and otherwise we have to resequence maybe, or add some sequencing to it, etc. But the most, let's say, optimal scenario would be to select from ongoing cohort studies and biobanks, which are there, which are rich with phenotypes. We do not ask for phenotypes at the moment, but we do ask you to choose your samples wisely from it, so from existing biobanks with lots of phenotype data, or you can start a new biobank yourself if you do not have such an infrastructure, and learn from countries who have done that already and see what is the best way to do that in the most effective way of doing that. So this is the layout of the program. Probably you will not be able to see it, but this was a calculation we did on this distribution of numbers per country, and then looking at the subpopulations, because we ask each country at the start, how is your population looking like? What kind of subpopulation do you have in your country? Like in the Netherlands, we have white Caucasian Dutch, but we also have Indonesian people, we have Suriname people, we have Turkish people, Moroccan people. What is the percentage-wide distribution? And based on that, we selected from all those countries in those tables, and you see them listed in each row. So the rows are all the subpopulations we uncovered in each of those countries. So for example, you see that Turkish people live in Germany, in Denmark, and the Netherlands. So if we all contribute a little bit of our Turkish genomes, we create a Turkish reference genome based on data from Denmark, Germany, and the Netherlands. That's how the Genome of Europe creates data from people not living in Europe, but who are healthcare consumers in the individual countries. So that's how it's distributed. We will revisit this table every so many months, especially this year now, to see how are the numbers working out now. A few highlights, I think I've mentioned most of them also. So we ask for minimal phenotypes, age, sex, and country of origin, that's it. But again, we ask you to choose your samples from existing biobanks and cohort studies, where we know there's lots of phenotype data, so we can go back to there. We're working mostly with short-read data at the moment, but we also have quite a substantial amount of long-read data from Oxford Nanopore and PacBio. We're going for a clinical grade genome, so 30x, so useful in clinical settings, also as reference data. And again, we allow various sequencing centers to collaborate and contribute to that, and that will lead to a strong infrastructure in Europe for that. Small word about the use cases. We want, of course, make a genetic map of Europe. How's the genetic diversity looking like in Europe through principal component analysis? A very simple use case, of course, the lookup of variants. So you do your family here, you see all these VASs in it. Well, let's have a look in the Genome of Europe database. Has this VAS seen before? And if so, where and under what conditions is this variant there? And we have some other use cases. We'll not go into detail there, imputation and polygenic risk calibration, but those will come later. I'll show you an example of what we do in Holland to contribute to that. So we have several epidemiological cohort studies in the Netherlands, in fact about 25 or so. So they are organized now in a so-called national consortium of biobanks and cohort studies that's funded, luckily, last year. We contacted them, say, okay, this is the plan, we have money for 5,000 genomes, which samples do you advise us to sequence? So they pointed, together with us, 3 cohort studies, the Rotterdam study, the Helios study in Amsterdam, and the Lifeline study in the northern part of the Netherlands. Those were the samples that will contribute to this first round of Genome of Europe. As I said, we currently have money for 100,000 genomes, but we want to, of course, sequence many more genomes. So together with them, we are selecting and picking the samples now from those existing biobanks, and those will be sequenced, in our case, in the Netherlands, by Erasmus MC with short read, and we do about 1,000 long read genomes, probably with Oxford Nanopore, but that debate is still going on. At the same time, we're talking to them about the governance of the data. So once we have those sequence data, where will it be stored and how will it be made accessible, not only for Genome of Europe, but also internally to those studies for individual investigators. And that's being done with, again, this NCC consortium, but also with what is called HealthRI, and that is a partner in the GDI project, the sister project in the 1 million genomes. So that's how we organize in the Netherlands the sequencing of the genomes, the availability of them for research and for application and contribution to Genome of Europe. We're also very happy in a special position that we have some special genomes also to be added, hopefully, because our royal family had their genomes sequenced. It was fantastic. But only part of their genome, by the way, it's not a complete genome. And they made a piece of art in one of the rooms in their palace where they receive guests. And all those bricks in the walls there are reflecting their genomes. See, these are actual genomes from both the king and the queen. Now, I'm an expert in genetics, but I have no idea what the DNA sequence is. So this tells you how sensitive this DNA information is and all the GDPR restrictions around it, because it's there out in the public, but nobody can understand. As you know, they receive very important guests there. For example, here, a guy you might well know from Ukraine. Kings are received there, people from NATO, bishops, religious people even there, very wise person from Canada was there, and also this guy from the US visited, was very happy. And I think one of the reasons he's now demolishing the White House is because he saw this room. He's saying, I want to have this DNA room in the White House. That's the real reason behind it. I was discussing just before that collaboration is not easy, because we have to collaborate in this project. And this slide illustrates you the type of prejudice we have to overcome. It's made by a Bulgarian artist, Svetkov. I strongly advise you to have a look at his website. It's hilarious. And he made this map of Europe, in this case, based on prejudice. And that's how it is in practice. We in Rotterdam think we can do things much better than them in Amsterdam. And we're both from Holland, yeah? And I think the same holds here from Poznan and Warsaw in Poland, I think, yeah. So you have to overcome those differences and start working together. Now, on science, this works quite well. I showed you the success of all the consortia, but politics is not always helping us. So, as you can see here, in Europe especially is blessed with many diverse countries and cultures and a rich history of changing borders and migrations, etc. So when I show this slide in the US, I usually call it Europe according to Trump, and people nod and they recognize this is how Europe is seen in the States. But on top of that, politics is not helping us. So we had Brexit, so they decided we should go separate, and suddenly we had a break with our closest friends and colleagues in the UK, whom we were collaborating. And well, Ivo can recognize this, Catalonia thinking of going independent from Spain. Well, you know, how will this work out? And then we have countries who really would like to join the European Union, but you know, there are doubts, are they really an important partner and should we allow that, etc. And big debate about it, so politics is not helping there. But I think we from science show how it can be done. So yes, we are collaborating with the UK, in spite of all the political differences and borders. We have good friends there, both in Genomics England and the UK Biobank. Also with the US, in spite of all the geopolitical storms we are in. Now, this is a guy I was lucky to meet from the good old days in the US, I should say. Mr. Obama started the precision medicine program, and we had a quick chat about what we're doing in Europe, and he was very enthusiastic about it, so that's great. More important is that Jeff Ginsburg from the All of Us program reached out to the Genome of Europe to collaborate. So, of course, US is a colony of Europe. We can call it like that, yeah? Here in this room. So it's very important. They have something to gain with the knowledge we generate here in Genome of Europe from understanding the genetic diversity in the US. So it's important to do that. That brings me to the closure, which is, please realize Europe is not limited to our continent here. We have a global footprint, yeah? We're everywhere on the globe. As you notice, I put a European flag on this country called United States of America, just to highlight that this is formed by European people. All of the European countries send people there. So it's a big mixture of European genetic heritage there. So that's why it's very important for them to be held by us, by the understanding the genetic diversity here in Europe, to understand the diversity in the States. But as you can see here, there are many small islands all across the globe where, especially through our colonial history, we have footprints, we have people still living there. And this is EU territory, yeah? So there's EU legislation working there. GDPR is active there. So we do have a responsibility to also reach out to those very tiny islands to help them being part of this project. Not in the current phase because there's lack of money, but this is one of the reasons to keep on sequencing, also including those countries here. So Europe is global, don't forget that. Finally, we're doing this now. Where are we going? We have new plans. I told you already about this new working group on pharmacogenomics, because I think this is a no-brainer. We should do that. And this is led by Jesse Sven and Lina Milani from Estonia and the Netherlands to work on that. So please join it if you're interested in there. The new plans for the working group 10, coming back where Genome of Europe got from, is to finish the job. The project is called 1 million genomes, so let's create 1 million genomes. We have now organized the infrastructure, we have all the people together, the experts, we can do this. This will allow to create a vast database of reference data, but also allow us to do big studies on genome-wide association studies to uncover European genetic markers for European diseases. And that can be, of course, then linked to activities to bring this into clinic and prevention programs, once we have those data based on European knowledge. It will also be, of course, very important to engage with the public, to inform them about these developments, to explain to them why it is important, how they can profit from this, and why they should support this kind of activity. And that, of course, will shift the activities, which probably you are aware of, which are currently mostly focused on care. So you see a patient, but that's too late, of course. Yeah? Then when the worst things have happened already, you want to prevent, of course. And in fact, as a doctor, you want to make yourself superfluous. Eh? No, not necessary anymore. That would be the ideal case. So what we need is less patients rather than more doctors. I stole this from Anders Metzploe, by the way. And I think genetic information can play a very instrumental and important driving role in this development for that. So that's why we are proposing now to Brussels this simple thing. We want to sequence 1 million genomes across Europe, and we want to genotype 50 million samples. You say, oh, that's a big number. Well, it's a small number actually, because we have close to 500 samples biobanked in Europe. And we know that through the BBMRI-ERIC activity. There are 500 million biobank samples in Europe. We're only asking for 10% for them to be genotyped, because we think that will be the basis upon which all these developments can play a role, not only for research and discoveries, but also for implementing this in clinical settings and prevention settings. And those are the listed activities we want to engage, but I informed you about it already, by doing all these discovery studies with it. Of course, we cannot do this in isolation. We want to collaborate. I showed you already there the map of the globe. We have a global footprint, so we should reach out to those partners. We also have to reform a little bit Brussels, because there are also silos there. You have those directorates. It's either Santé or Connect or RTD, and they're not talking to each other. They sometimes fund the same thing twice. We want to prevent that, be more efficient, and we have to closely collaborate with industrial partners to make this work. And I hope those will be European industrial partners, not US or UK-based European partners. So with it, I'd like to thank you for your attention, and not without saying a big thank you to my group, because I'm simply standing on the shoulders of them, just telling them about what they are doing. And especially a big thank you to all the people involved in Genome of Europe. It has been a fantastic experience to work with you. I got a lot of energy from it, and I notice you also. So I'm looking forward to the coming years of collaboration, and including welcoming new partners like Poland in this. So thank you for your attention.
Thank you very much, Andrew, for this fantastic talk. I guess to keep us on schedule, we have to move on, but if you want to exchange a couple of words with Andrei, there will be the opportunity during the coffee break, which is in a couple of presentations ahead of us. So thank you again. And now let me introduce our next esteemed guest, Professor Jan Lubiński, who is a world-class authority in clinical genetics and a true pioneer in the prevention of early detection of hereditary cancers. Professor Lubiński focuses on translating genomic discoveries into life-saving clinical practice, particularly through his groundbreaking work at the International Hereditary Cancer Center in Szczecin, and recognized as a global leader in identifying genetic predispositions to breast, ovarian, and colorectal cancers. His innovative research on the role of microelements had set a new standards in personalized cancer prevention and patient care. So, Professor, we look forward to your presentation, and please lead the way.
Thank you very much for such a great opportunity, to get first invitation from Professor Henschuch and Professor Legocki, actually, I think. And it is my honor and pleasure to have this lecture in front of you. I was asked to provide a presentation on genetic basis of cancer and to show results of long-term studies in Poland. These days, actually without deep sequencing on large scale, but with good basis to make it in the near future. It's not bad to read literature, and making it, I found in the late 80s strong data suggesting occurrence of high-risk genes for common cancers such as breast, ovary, and colon. The main group which was presenting this data was American one, where head was Henry Lynch. I remember that when I was presenting the first data about such phenomenon like hereditary cancers, people were just laughing at it. They did not believe. After a few years, we found that it is a good direction of studies. We created the first in Poland cancer genetic outpatient clinics. It was 1992. And what important, we understood that we need to create network. Nobody believed it, so Poland was like a vacuum. And we established cancer genetic outpatient clinics practically in all of regions of Poland. It was late 90s, supported also by European project on development of cancer family syndrome registries in Eastern Europe. For 11 countries, 13 centers, including Dutch. And now we have not in infrastructure. We have a biobank with 600,000 persons, from whom we have blood, DNA, from cancer patients, relatives, and from controls. We have registered with more than 30,000 families with cancer family syndromes, with strong aggregation of cancers and biological material available. Also, some other very important elements of infrastructure we developed. First, a national program for management in families with high genetic predisposition to cancers, which I wrote in 1998, and which is active, very active till now. Center, special center just for diagnostic and treatment of hereditary cancers. Also companies. Some projects are much easier to realize with contribution of companies. Some projects are much easier to realize with university money. Having this infrastructure, it was possible to achieve a series of discoveries and applications of these discoveries. First, we identified the genetic homogeneity of Polish population. What made that DNA testing based on founder mutations have been simple, cheap, and fast. Very important discovery, BRCA1 gene, just 3 mutations dominating in more than 80% of families, of Polish families, with strong aggregation of breast ovarian cancers. Right now, these days, we are working with basic tests using just 5 founders. What is discovering more than 85% of Polish BRCA1 carrier persons. Another gene, CHEK2, also just 3 protein truncating mutations, strongly dominating Polish pattern of CHEK2 high-risk variants. PALB2, years between the first discovery in 2000 about BRCA1 and PALB2, there is 15 years of difference. It takes time. You need to study large numbers of breast cancer patients, of controls, but finally we are achieving such data like here, publication from Lancet Oncology, 2015, just 2 Polish PALB2 mutations and there is 100,000 of Poles carrying this, one of two. And the final, fourth Polish gene, I would say, NBN. We have one founder mutation, NBN deletion 5, and associated with genotype GG. And it is associated with more than 4 times higher risk of prostate and also breast cancer. Summarizing this part about founders and testing, we have just 11 high-risk mutations in genes BRCA1, PALB2, CHEK2, NBN, which are associated with high or very high risk of cancers, and these mutation variants are carried by around 1 million of Polish inhabitants. It is matching to this what you said just a while about finding for variants characteristic for a given population. No doubt that this is a huge chance to be focused on different ethnic groups. And this program from '98 about management for hereditary cancer is assuring very high standards in diagnostics, prevention, and treatment. Beautiful example. The first Polish Lynch syndrome family, asymptomatic colon cancer, found in this lady. And then she went for colectomy, so different treatment than for standard one. Preventive resection of uterus, fallopian tubes, and ovaries. And she is alive in 2025 with good health status. We can say 30 years of life for free. Her mother died at the age of 40. Her sister died at the age of 39 because of colon ovarian cancers, and she is saved. Now, look, having this, we can say, not genome, but genes-focused program is very efficient in treatment. Look, results of treatment of Polish women with breast cancer at the age less than 40 is better than American one. Here we have 10-year survival, 77.9%. In white American, 75%. In black American, 62%. In Canada, 59%. And these are not numbers which we created. These are numbers which have been created by Toronto University. And now, there are several reasons why it is better, but one of them is that, look, average size, tumor size of breast cancer in this group in Poland, it is 21 millimeters at the time of diagnosis. In States, among whites, it is 29 millimeters. So having information coming from genes and by development of special surveillance program, actively following these persons, you can achieve such efficient screening programs. We made population programs in West Pomerania, for example. We collected by family doctors information about cancer family history from 1,200,000 individuals. In all of them, if there was a breast, ovarian, or colorectal cancer among relatives, we performed BRCA1 founders and also founders for HNPCC genes. And we repeated these activities using several mass media, like this action with Journal for Women, your Fashion, or directly another journal, very popular in Poland. This was population testing for CHEK2. And now we have, after all of these actions, we have probably the largest world registries for at least BRCA1 founder mutations, more than 10,000. People registered and under surveillance. And the same story with CHEK2. Having such registries, you can study several aspects, several clinical aspects. Like here, there's JAMA oncology paper. 11 countries, 59 centers. This is coordinated by Toronto. And to look on contribution. Canada, Poland has the largest contribution, more than other countries. And we are coming to very, very important conclusion that if you put BRCA1 carriers on the surveillance program with MRI, then breast cancer mortality is lower 5 times than in subgroup without this program. 5 times less deaths because of breast cancer. This is a great achievement. Another story. We were the first to show that platins are critical in the treatment of breast cancers associated with BRCA1 mutations. Here you can see just 12 cases and publication in Journal of Clinical Oncology. Why? Because out of 12 of these women, 10 showed complete pathologic remission. And this is the best symptom of perfect prognosis for survival. Actually, if you use platins, which is this black line, and you combine it with oophorectomy, 10-year survival is higher than 95%. So practically they can be treated. In the past they were dying without hope. And finally, some achievements in new genes of high risk for breast cancers. Everything is based on these registers of this biobank. Nature Genetics publication, we showed that RECUL mutation, it is a new gene of high risk for breast cancer. Sorry, maybe I will mention. This was done in collaboration with French-Canadian data. Why? Because in both populations we have founders. In Poland, we have just one founder present in 30 breast cancer patients out of 13,000 unselected breast cancer cases, and in 2 persons from 4,700 controls. Similar data in Quebec. And the last, and the second gene, new gene, it is ATRIP. The same story, whole exome sequencing, identification of founders, and association studies on Polish population and UK bank participants. And the final achievement coming from these huge registers. This is project which we call Selena. This is a project where we optimized selenium and arsenic blood levels in females from families with hereditary breast cancers. Basic findings for this interventional project. All-cause mortality, data from the US, prospective observations of 18 years in almost 14,000 adults, and you see that we have U-curve. The lowest risk of mortality is in states when selenium blood level is around 140 micrograms per liter. In Poland, we have similar data, similar U-curve, although the optimal blood selenium level in Poland is 100, not 140. Different population, different environmental exposure, and probably genes as well. Another basic finding for these studies was correlation which we found between risk of cancer and blood arsenic level. If this arsenic level is not too high, there is up to 10 times higher or lower risk of being affected by cancer. This is on females without BRCA1 mutation. In BRCA mutation carriers, you have a similar story. All of these correlations are not so strong. Out of 651 carriers with low arsenic level, you have 32 cancers. Among 650 carriers with high arsenic levels, you have 71 cancers, more than 2 times more. So we recruited more than 7,000 females, BRCA1 mutation plus or BRCA1 mutation minus, different arms. All of women got suggestion of lower of arsenic-free, almost free diet, if blood arsenic level was above 0.6 microgram per liter, and then selenium recommendations depending on whether it was excess or deficit. We had placebos, selenium supplement, diet intervention, or just observational controls without any recommendations. And results: BRCA1 plus group. Selenium and at the same time arsenic cannot be too low. If it is the case, the risk of cancer is higher, almost 5 times. BRCA1 minus group. If you have optimal selenium level, 98,108, and your arsenic level is not too low, but 0.58, 0.75, you have almost 5 times lower risk of cancer and strong tendency for lower risk of deaths. We found that there is synergies between selenium and arsenic, so we introduced risk index, which you can calculate in such a way that you are taking selenium blood level plus arsenic blood level multiplied by 50. And then you see that this correlation between selenium and arsenic levels is stronger. If index is 125, 145, your HR is 6.5 times higher. Your risk of cancers is more than 6 times lower if your index range is 125, 145. Very similar strong tendency for deaths. In BRCA1 plus, index of risk index optimal is 120, 135, and if you compare 120, 135 versus other values of index, it is almost 6 times lower risk of cancers. So, in other words, directly saying, your risk in BRCA1 carriers is roughly 80%. In this subgroup with appropriate selenium and arsenic, after intervention, it is 6 times lower, so it is 15%. Huge, huge difference. There is summarization of these optimal levels. We can conclude optimization of blood levels of selenium and arsenic is attractive option for women from cancer genetic outpatient clinics. We have, in reserve, we have genotypes. Look, BRCA1, catalase, and arsenic should be low. Catalase genotype CC, selenium should be high. HRG gene, this is histamine-rich glycoprotein. Risk index should be between 125 and 170, and the risk is 5 times lower. BRCA1 caspase, non-CC genotype, and selenium cannot be low, should be higher. Summarize of this part. Optimal effects of selenium and arsenic on cancer risk and all-cause mortality can be achieved by supplementation or diet intervention after analysis of DNA for genotypes of catalase, caspase, and HRG, and obviously blood levels of elements. The last part, elements and prognosis, survival in cancer patients. This is a historical slide because this is the first demonstration that serum selenium level is strongly associated with breast cancer patient survival. This is 10-year survival, and you see that in the first quartile with low selenium level, you have 46 deaths. In quartile 4, serum selenium level above let's say 95, just 16 deaths. I'm very glad that it was validated in Swedish population. By the way, the Swedish researchers are very elegant. The only previous study on serum selenium breast cancer mortality is study by Lubinsky et al. It's very rare that somebody is making such statements. Now, actually, we deeply believe in this correlation. I was showing you data on quartiles, but subjectively, you can identify particularly low or optimal levels of selenium, and then this difference is very, very strong. If selenium level is less than 70, every second breast cancer patient is dying within 10 years of follow-up. If selenium level is around 100, only 2 out of 64 breast cancer patients are dead. Very strong data also for prostate cancer. Selenium and zinc we studied. Let me show you here. If selenium is the first quartile, zinc is the first quartile, there is 27 deaths and 22 alive patients. If selenium is quartile number 4 and zinc number 4, there is just 1 death and 27 prostate cancer patients who are alive. HR is 20. It's unbelievable, but I'm sorry, we have such data. In general, elevated selenium and zinc levels can predict a favorable prognosis, whereas high copper levels predict a poorer prognosis for all studied malignancies, cancer of the breast, prostate, colon, especially Lynch syndrome cancers, lung, larynx, pancreas, kidney, and malignant melanoma. In all of these cancers, these correlations are going the same direction. And at the end, acknowledgements. I calculated one day that we have been working all together more than 400 persons. Certainly these are not all. And my beloved special persons who really contributed a lot in our work. Professors Domagała, Wojciech Koprowski, Kay Hibner, Lynch, Zurhausen, Harald Zurhausen, Nobel Prize for HPV, Rodney Scott, Stephen Nerot. I am making summary. This series of presented genetic clinical correlations need further investigations to improve sensitivity and specificity. And very critical, absolutely, I totally agree, future studies based or including analysis of genomes. Thank you very much.
Thank you very much for this great presentation. I think we can move the discussion for the coffee break. We can make it maybe a little bit longer for this purpose. And now, in this session, the last part will be the presentation given by Professor Hanshu. You already seen her, met her, and heard from her. She's the director of the Institute of Organic Chemistry of the Polish Academy of Sciences in Poznań, and she's a leading expert in molecular biology and genomics. Throughout her career, she has spearheaded groundbreaking research on human genome and coordinated genomic projects and coordinated, among others, the Genomic Map of Poland. And today, she will present the core foundation and strategic goals of the project. That is the reason why we meet today. And the project will bridge the world-class laboratory research with its vital impact on public health and diagnostics. So, Professor, please take the lead.
Okay, thank you very much, Gosia. So the last presentation before the coffee break. Maybe the coffee break will be even longer. We'll see. So usually this is the last slide of the presentation, but I decided to start from this, and maybe I even should do it at the beginning of the meeting to tell some few words about funding, because it's important. So the funds for the project Genomics for Poland comes from the European Funds for Smart Economy program. It is in Polish Fundusze dla Nowoczesnej Gospodarki, Europejskie Fundusze dla Nowoczesnej Gospodarki. And in fact, it's continuation of 2 earlier programs, which was called Innovative Economy and Smart Growth. And in both of these programs, we also participated in calls, and we got some funds for our previous projects. This particular call was within the Action 2.4, which is called Smart Economy Research Infrastructure. And it was a special call because it was called for so-called network organizations. So it's, in fact, we are going to make a network during the project, but it was also an obligation to be a part of a network to apply for the project. And our network was Polish Academy of Sciences, because 3 institutes of Polish Academy of Sciences, for those people who are not, who don't know that, I just want to explain that Polish Academy of Sciences have 68 institutes now. So at least 2 participants, at least 2 representatives of the network organization was obligatory to apply for the project. We decided to include 3 institutes and also some other units. I will tell you more later. So the source of funds, this Ministry of Funds and Regional Policy, Ministerstwo Funduszy i Polityki Regionalnej, this is the managing authority, but the responsible organization which plays a role of, let's say, intermediary institution is Information Processing Center, National Research Institute, which is known as OPI. And the project value, as you can see, is quite high. It's almost 250 million zlotych, but it's not it's a final value of the project, but in fact we got funds for 180 millions. The rest has to be contributed by, as an our contribution, financial contribution. And the project will start January this year, and it will take 4 years, so till the end of 2029. Institute of Bioorganic Chemistry is a leader of the institute, a leader of the project. As a representative of the Polish Academy of Science, it was also obligatory that this representative of the network should be a leader. And fortunately, I am the director of the institute now since 2 years, and I'm also the head of the Laboratory of Genomics, so it's really my project, and I'm really fan of this. I'm really glad that we can start this initiative. In fact, 12 units are engaged, so 3 institutes of Polish Academy of Sciences, 6 universities, 2 national research institutes, and 1 Łukasiewicz Research Institute. I will show it also later. And it's also important information that non-financial contribution to the project is thanks to Bicoders and Analytic Genetica, so 2 companies, Polish companies, which decided to support our projects. So thank you very much. So nobody has doubts, I think, after the first 2 presentations, that genomics was a real breakthrough in our research, because in fact it allowed us to change the scale. So from single gene level, we could transfer to the genome scale. So it's completely different research level, completely different understanding. So we do not focus on single gens, single mutations, but we try to make more holistic view on the whole genome, and not only one genome, but also a lot of genomes, so the population. So from partial information, we can got complete information, but still it's very complicated. So we have to learn much more to understand the information which contains our genomes. Human Genome Project, of course, it was, without this project, our project would have no sense, and other projects like Genome of Europe, yeah? It was 13 years, a very expensive project because more than $3 billion. But in fact, when we think about benefits, they are invaluable. So 13 years of the first, let's say, complete genome, but in fact, the research was going on still. So in 2022, we had telomere to telomere, so T2T consortium complete genome. So still research is going on. And here I would like only to present a few faces of the Human Genome projects. They are only males. And 2 of them are Nobel Prize winners. None of these Nobel Prizes was for human genome sequencing. Yeah? It was not awarded. However, it was a very important initiative. And James Watson, who died November last year, he was not only very related to this initiative, but he was also a DNA donor. So he donated his DNA, and he was one of the first individual genomes came from James Watson, so the Nobel Prize winner for the discovery of DNA structure. John Salston got the Nobel Prize for the research of apoptosis in C. elegans. But generally, all of these people, some a little bit controversial, they contributed much to the Human Genome Projects and many, many other people all over the world. Thanks to the Human Genome Project, we have now reference genome, and it's gold standard. So each newly sequenced genome which we sequence is compared to this reference. It's for all bioinformaticians, it's obvious that it's GRCH38. We have now version P14, which is available from 2022. And it's still, I think, the most commonly used reference in human genome research. Impact of genomics is definitely huge, nobody has doubts. So the development of genomics impacted our knowledge because we know more about worldwide genetic variability. We know more about molecular basis of the diseases. Of course, genomics contributed to the development of science, so new discoveries, research and development projects, new tools, methods of analysis, but it also contributed to healthcare, so cancer predisposition screening programs, newborn screenings, prevention, diagnostic therapies, and so on. But in fact, also it was very expensive. The Human Genome Project was very expensive. It made huge profit in money, so it stimulated market and business very strongly. So there are many entrepreneurs and many business companies which started to act in genomics field, and I think it's still they are still going quite well. For example, Illumina, everybody knows, yeah. But also genomics stimulated industry development, especially pharmacy and biotechnology, and also it impacts the society. So we are considering ethics issue, our origin, our identity. So all of these issues are thanks to the genomics, which was developing so fast and dynamically within the last years. Genomics in other countries is quite advanced, I would say. I think the most prominent examples are UK and USA. So in UK, the 100,000 genome projects, now it's more than 500,000 genomes, which are, the results are already published. And in the All of Us research program, which was launched later, but still they are quite advanced. I think that they got quite good funding from the National Institutes of Health, and they have collected already more than 600,000 participants, and also databases about phenotypic data. So it's very, very valuable data sets, and all of the researchers all over the world can use these resources. This is the simple National Genome Project timeline I prepared. Probably it's not possible to show all the National Genome Projects on the one timeline, but I tried to put as much as possible. And you can see here that Iceland, they started very early. So 1996, they started to sequence their population. And also Andreas showed a slide when you can see that more than almost half the population is, or almost all population of this country is already sequenced. So it was the pioneer, in fact. But also there was a small project called Sardinia. So one island of Italy was more than 2,000 whole genome sequence, quite early. And Estonian Genome Project, it's a very, very valuable project. I really appreciate because it's a small country. And here you can see Andres Metspalu. He is also the participant of Genome of Europe. Now is responsible for communication and dissemination package. But in Estonia, it was a fantastic environment. So the government understood very early that genomics is important, and they decided to give some funds and to collect samples and start sequencing quite early. And I think that it's in education, and Andres Metspalu also always repeats this, that education, social education is very important because it's important to persuade people, society, that they should not be afraid of such testing, that they should participate, they should be curious because it's really interesting. and they did it really well in Estonia. So I think it's more than, they have already collected more than 200,000 whole genome sequences, which is about 20% of adult population of this country. So it's really, really impressive. And then you can see quite a long gap. So from the early of this century and till 2015 maybe, where there started a boom of national genomics project. But in fact, the gap was not really empty because it was the time of recruitment. So many projects started from the first phase, where they just decided to collect samples to support a biobank collection. So they started from the samples, biobanking, and then decided to apply for the projects and funds for sequencing. And in the meantime, the cost of sequencing, human genome sequencing, decreased. So maybe it's good that they wait a little bit longer, because now they can make more for the same money. And in fact, many countries have their own programs for genome sequencing. Many different countries like Korea, Qatar, Emirates, Hong Kong, Singapore, China, of course, Finland, New Zealand, Turkey, and many other countries. And of course, you can see also here a Genome of Europe, which is a more international project. And as Andres said, that the plan is to sequence 100,000 samples, but probably it will be more. So we can see here also similar information, but presented on a map. But it's not the percentage of population, but it's the number of genomes which are already sequenced or which are planned to be sequenced. Because there are two countries which have ambitious plans to sequence at least 1 million genomes. It's Taiwan and United Arab Emirates. So they have very ambitious plans, partially completed. Then there is a group of countries which decided to sequence between 200,000 to 600,000 genomes. And it's Estonia, Finland, UK, and USA. They are completed this plan, and probably they go on and they sequence more genomes. But the plan was completed. France also is going to do it, but it's not ready yet. And there are also some countries which have planned to sequence 100,000 genomes, so like Genome of Europe, but also Saudi Arabia, Turkey, Singapore, Mexico, Egypt, or Canada. So still, when we think about genomics in Poland, I have to admit, we are still far from the others. So the current number of whole genomes available in Poland, and also available in our consortium, is about 11,000 sequences. So it's not a very huge amount, you can see. It's only about twice the number collected by the UK Biobank in the pilot study between 2014 and 2016. And this equates to just 1.7% of the entire USL all of us collection, which is the largest cohort now. So it's really not very impressive what we have, but we try to go on, and maybe our project will help a little bit more to be better. Some countries have collected really huge genome sequence cohorts. And what is important, national genome projects, national genome sequencing projects are often supported by governments, which guarantees funding, data maintenance, and security. And here in Poland, we don't have such situation. I think maybe in other countries too, but I think that our politicians do not understand that genomics is significant, that it's important, and I would like to persuade them, but nobody came here, so I had no opportunity to do it today. Maybe I have to go to them and directly try to discuss. There is also, what is another obstacle, is a lack of understanding at the level of politicians, decision makers, and society. So in the Polish society, which is very traditional, conservative, it's sometimes very difficult to persuade people just to donate their DNA. They are afraid of many things. Yeah, they are afraid that we can use it for other bad purpose. I don't know, but it's really difficult when we were making our genomic map of Poland. I was involved in collection samples all over the country. And in some regions, it was really difficult. For example, Highlanders, they were very opponent. Yes, they were very afraid of donating their samples for such a research project. And of course, there's a lack of standards in genome sequencing technology. There are no standard protocols. There are no legal and ethical issues. There are no rules, I would say. And the maps of shame, I have to show. One, it was shown already by André. So the European initiative, 1+ million genomes, 1+ MG. Poland is still an observer only. So that's why we cannot be a fully partner of the project such as Genome of Europe. I really regret, and I try to discuss about these things whenever I can, but it's not so easy. And the 1+ million genome initiative started 3 projects. It was beyond 1 million genome project, which was a kind of preparation for genome sequencing in Europe, and then GDI, so European Genomic Data Infrastructure project, which prepares everything from the infrastructural point of view, and of course Genome of Europe, which is the project for data collection, which is going now. Our project consortium contains 12 units, as I mentioned at the beginning. The Institute of Bioorganic Chemistry is a leader, and an important part of our institute is Poznań Supercomputing Networking Center. So it has a quite huge role in this project. We have also Institute of Human Genetics, Polish Academy of Sciences, Nęcki Institute of Experimental Biology, Polish Academy of Sciences, Poznań University of Technology, so our informaticians, we collaborate for many years. University of Warsaw, especially Center of New Technologies, Medical University of Białystok, University of Łódź, University of Gdańsk, Jagiellonian University, Cardinal Stefan Wyszyński National Institute of Cardiology, National Research Institute in Warsaw, Maria Skłodowska-Curie National Research Institute of Oncology, Gliwice branch, and Łukasiewicz Research Network, Polish Center for Technology Development. You can see that we come from different cities of the country, so we have quite a tight network within Poland, and the different partners have a little bit different competencies. So some are more developed equipment. The others have quite good biobanks. So I think others are bioinformaticians. So I think altogether we can make really huge progress. I told that the Poznań Supercomputing and Networking Center is a part of our institute. It's affiliated by our institute, and it's in fact half of the institute. So the whole institute employs now about 800 people, and half of them belongs to PCSS. Yeah. And what is important that PCSS is the unit, is the center, infrastructural center, which developed Polish optical internet. So it's called Network Pioneer Network. So it connects all the cities, all the research units, which are all over the country. So we are very good connections. So our network in the projects also overlaps with the network of these connections. The main goals of the project are to establish a nationwide network of laboratories which are conducting research and also providing services in the field of genomics, so serving academic, commercial, and healthcare institutions. To build and develop advanced infrastructure for genome research, including machines for high-throughput long-read sequencing and creating optical maps. And to support the development of science, personalized medicine or precise medicine, diagnostics, innovative therapies, and targeted prevention, what is more for future. The idea which stands behind the project is we would like to transfer from traditional towards more personalized or precise medicine, so to focus on patient, to take into account the genetic background and all other features we can know about this patient, make it digital, and draw some conclusions also based on genetics. Rare diseases, it's also a very important issue in our projects because they are still underdiagnosed. There are many rare diseases, but the cases of particular disease are not very frequent. And here we plan the collaboration with the National Institute of Geriatrics, Rheumatology and Rehabilitation, and they have already declared during when we applied for the project, they declared our collaboration, and they are able to share with us the collections of ultra-rare diseases. So it's very important for us because 4 years, maybe it's not short time, but it's definitely too short to collect rare disease collections. So thank you. So more detailed aims of the project are as following. So as I said, we wanted to create a network of collaborating collaborators, because it's not true that genomic research was not done in Poland. It was done before, but it was rather dispersed. So single units, single research institutes or universities made some projects connected with genomics, but it was dispersed. They were not collaborating, sometimes even competed. I would like to collaborate. I think it's better to collaborate than compete. So, and we would like also to have our laboratories accredited. So we are going to apply for the ISO norm to be the laboratories which can which can offer really professional services. We would like to supplement key infrastructure resources. They are not bad, as you can see in a minute, but we need more, especially the latest technological solutions. The market is very dynamic, so it changes quickly, especially in long-read sequencing technologies. We would like to create a national genomic data repository. So the one place where all the sequences from Poland will be collected, also from the previous projects, previous studies. Probably there will be a need to reanalyze this data because they were analyzed in with different methods, different pipelines. We would like to archive all the existing data from various centers. We also are going to collect new genome collections, so particularly from rare and pediatric diseases, and developing a Polish contribution to the Genome of Europe project, because we don't have the money for sequencing in the Genome of Europe project. What we are going to achieve also, of course, it's not only up to us, but we will try to achieve Poland's full accession to the European Initiative 1+MG. We would like to prepare our infrastructure to be connected with the European infrastructure, which is also important to transfer the data between research units all over Europe. We would like to develop national standards for acquiring, processing, and sharing genomic data, developing also a software for categorizing genetic variants, including variants in non-coding regions. We have some experience in this area, so we would like to include it into our author software, which will be helpful in prioritization of variants. We would like also to develop some genomic data processing pipelines, and what is a very, very big challenge for us, we would like to use quantum computer and to maybe use it to apply it for genomic computation, which is still completely empty area. Nobody does it yet. Yeah, so we hope maybe we'll start it. I hope so. We would like also to use artificial intelligence to data analysis, deposit new biological materials in biobanks. Maybe we will be able to collect more samples than sequence, so it's better to secure this material. And of course, the last thing is to develop some guidelines for establishing ethical and legal standards for generating and processing genomic data in Poland, and also to make some educational activities to get better understanding in the Polish society. So G4PL work plan is quite basic. There are only 4 work packages. So work package 1 is just biobanking. Work package 2 is infrastructure for genome sequencing. Work package 3 is infrastructure for data analysis. And work package 4 is infrastructure for archiving and sharing data. So all of these work packages are quite big, and they will be interacting because otherwise it's impossible to make such a project. And when we are thinking about biobanking, we would like, first of all, to take advantage from the existing infrastructure. So we are not going to build new biobanks, but rather use the biobanks. They are available in consortium members, because our consortium partners participate in the Polish biobanking network and also the national node of European BBMRI ERIC infrastructure. So we only plan to supplementation with small equipment, and of course archiving samples collected with the project, and maybe do some external services, because of the network of laboratories we are obliged to offer some service to the external environment. Work package 2 is infrastructure for genome sequencing, and here I present the current state on the level of whole consortium, so you can see it's not bad. So we have many sequencing machines, mainly in the short-read sequencing technology, which is offered by Illumina in the market of genome sequencing. But only two now, NovaSeq X Plus, so the most high throughput sequencing machines which allow for sequencing within 1.5 day, 124 samples, yes. So it's the most high throughput machine, and so this sequencing using this machine is really cost effective. So it's the best choice when you are thinking about sequencing human genomes. So we have only two now. One is here in our institute, the other one is Białystok. But we are planned to buy at least 3 more, so it will be more capacity. We have also, of course, smaller equipment which is necessary, so automatic pipetting stations and some other instruments like bioanalyzers to control the quality of libraries before sequencing. For long-read sequencing, we are not so well equipped, so we have, in the whole consortium, we have 3 instruments from nanopores, not very much. And we have one PacBio, but equal to E instrument, so it's rather expensive to sequence within this instrument. We will have to buy a new one, which offers some better solution. So this PacBio equal to E is in our institute. We have also access to single cell technologies in our institute and also in few other consortium partners. So what we plan to buy, we want to supplement the equipment for short-read sequencing. As I said, we would like to buy in total 4 high-throughput sequencers, and at least 3 of them will be like NovaSeq X Plus, or this capacity. We are also going to purchase of equipment for long-read sequencing because we do not have enough and different platforms for this third generation sequencing technology, and also to buy equipment for optical genome mapping, which is not available in the consortium yet, and it's very useful for identification of large structural variants in the genome. And of course, some additional equipment such as pipetting stations, DNA analyzers, mass spectrometer for DNA methylation analysis, and many computing stations and server, because we also need more computational power and more space for archiving data. So our future capacity per year should be about 60,000 human genomes with 30x coverage in short-read technology, a few thousand whole genome sequences with the use of long-read technology. And we plan to get about 3 to 5, maybe 6 petabytes of data storage, which is more or less equivalent to data obtained from the sequencing of 30,000 to 50,000 of whole human genomes. Generally, we plan to build infrastructure which is sufficient to cover the entire country genome sequencing needs now. Of course, the needs can change in future, but now I think it would be quite enough. And it will allow us to do all the sequences we need in Poland, in the country. We won't have to send it anywhere abroad. So I think it's worth it. When we think about infrastructure for genome sequencing, we also think about new sample collections. So we generally plan to sequence about 6,000, maybe 7,000 samples, maybe more. It depends on the price of the sequencing. We will negotiate. But including these samples, we will have a medical data set, some from patients, because we have some clinical units in our consortium. So we plan to get some sequences from patients suffering from rare diseases, but also including neurodegenerative diseases with Tourette's disease, with repeats of trinucleotide sequences. They need definitely to use long-read sequencing technology, because usually the repetition region is longer than the short read, which we can obtain from the Illumina sequencer. We also would like to sequence samples of DNA from patients with metabolic, hematological diseases, rare cancers, and cardiological diseases, because we all have such possibilities in our consortium. But in fact, we plan to sequence up to 7,000 samples, but you have to remember that to catch a single case of ultra-rare disease, we should sequence at least 100,000 genomes and have luck, yeah. So it's not so easy, and probably we still just make a very short, very small. We cover a very small variation of our population, even if it's rather homogeneous. We also plan to collect population data set, so the representatives of Polish population, including representatives of minorities, so to collect a new data set as a Polish contribution to the Genome of Europe. And we have to remember that the contribution of particular minorities fluctuates. And since the war in Ukraine, I think, I checked it. It's the most numerous minority in Poland. It's now Ukrainian minority. So when we think about sequencing of genomes of Poland inhabitants, we have to take into account that 50 Ukrainians should be sequenced for every 1,000 samples from Poland, to reflect the real structure of our population. What is also important, we are planning to do at least 2,500 long-read sequences. So it's quite a good, I think, achievement if we can do it. Work package 3 is infrastructure for data analysis. So it contains aggregation, standardization, and analysis of data, comparative analysis of data coming from short and long-read sequencing. It's still not very standard. There are not very standard procedures how to deal with it. I said that we plan to develop our own software for variant analysis and prioritization. There are some softwares available in the market, but you have to pay quite much money for it. But we also wanted to include our knowledge and experience. It's the head, Professor Piotr Kozłowski, which is the head of the Department of Molecular Genetics in our institutes, has a very good experience with non-coding regions, microRNA coding genes, and also the mutations which appear in these regions can also be can have potential of pathogenicity. The plan is to implement AI tools in data analysis, elaborate new analytical pipelines, visualize data, and application of quantum computing for genome analysis, which is the highest, I think, challenge in the project. So we would like to maybe prepare the environment for such computation, and probably it will be hybrid computation. So we would like to connect a quantum computer with traditional computational resources. And the last package, which is called Infrastructure for Archiving and Sharing Data, Poznań Supercomputing Networking Center will be responsible for this package. And fortunately, we have access to quite good computer stations in PCSS. But still, we have to increase storage capacity. We plan to make a central data repository. Of course, it will be in 2 copies, and another copy will be in another localization. The data sharing platform is network with graphical interface and user-friendly interface. What is also important, we have to ensure data security. And of course, everything we are going to do should be according to the procedures, according to the compliance of the GDPR and FAIR, so the data should be findable, accessible, interoperable, and reusable. We also would like to concern some legal and ethical issues. And what you can see here on the picture is first quantum computing in Poland, but also in Europe. It's one of the 6 quantum computers which are developed in Europe, different technologies. And here we have technology, ion trap technology. So we hope we can use this machine also for genomics. It will be the first case, I think. And also what is developing now in PCSS, it's Piast AI, so it's the AI factories. So they will develop the space for data analysis, which is necessary for when you want to use AI algorithms. So Thanks to the PCSS, we have access to these resources and capacity, and the capacity will be even larger after this project. So when we think about future, the research objectives of this project is solving important biological problems, such as understanding the sources of genetic diversity and the molecular cases of diseases, development and implementation of genomic-based diagnostic methods, particularly for rare diseases and cancer, searching new therapeutics targets and developing new strategies of personalized therapies. We do it in our institute, but maybe it will be more possible for a larger scale, the scale of the country. Developing AI tools for scientific purpose, big data analysis, and also clinical purpose to enable personalization of therapy and prediction of treatment response, and also to prevent, because it's definitely better to prevent than to cure. And at the end, yes, development of application for the analysis of genomic and other biomedical data, also from other medical tests and imaging using quantum computers. So finally, I would like to tell that Genomics for Poland is really for you. So it's not just a noble slogan, because we build the infrastructure for the scientific community, not for us. We don't have such big needs, yeah? So we do it for the environment. For scientific community, healthcare providers, also for business sectors. So we are already offering some services, but I think it will be possible for a larger scale in future. We are open for collaboration as well as commercial services. And we encourage people who are not consortium partners to become users of the infrastructure, because many people came to me and they asked why you didn't think about us to include us into the consortium. Yes, I know that maybe a consortium could be larger, but in fact, we don't build it for us, and we have to offer to other units. So I hope that the other collaborants, potential collaborators, can be user of our infrastructure. So it's not only for us, yeah. So it's also for you. So we really encourage to collaborate, and we also have contact for potential DNA donors. We of course do not have the opinion of Bioethical Commission yet, so we will apply submit the application. But in the meantime, we can start collection of. Declaration, I would like to participate in the project. So there is an already active email, G4PL, starting, and anybody who would like to donate his or her DNA sample, please do not hesitate to write to this email address. And at the end, I would like to thank a few persons, especially Natalia Koralewska. Is Natalia here? Can you show us? Natalia is the person. Natalia is the person without her help, I'm sure that the project would not be completed, yeah? The application. So he was the biggest help for me. And it's very complicated. All the consortium partners are aware of the fact how complicated the application was. And Natalia was the person who was able to be with us and to be our guide, because she has experience before, and unfortunately she has no time to be engaged much in our project. So Gosia Marcinkowska-Swojak will play her role, I would say now. But Natalia, definitely thank you. Thank you very much again. We also have very huge contribution from our scientific secretariat. We have a small scientific secretariat, it is called, but there are 10 persons who are just helping, writing grants and everything which is connected with grants and organization of grants and reporting and so on. So Marcelina Wysocka and Paulina Szatkowska, are you here? So, okay. So they are very competent persons, very patient. They spend a lot of time to complete the application and make correspondence with all the participants. And so thank you. Thank you again. And I hope you will be with us till the end of the project, at least. We have also very strong support from our finances. So Tomasz Kulpa, is Tomek here? Okay. Tomek Kulpa will take care of our finances in the project as a coordinator, but also he has support of our Deputy Director for Financial Affairs, Adam Kubasiński. He's probably not here because he's a very busy man, but he also helped at the... And I didn't mention about it, but we also include some works which are collected with preparation laboratories and rooms for the new infrastructure. So we also need quite strong support of our engineer, and Marcin Zieliński is also very helpful in this project, so thank you. And of course, the project would be not possible without significant contribution of Poznań Supercomputing Networking Center. So I thank you, Robert Pękal, who is the head of this unit. But also Tomasz Piątek and Tomasz Kuczyński. Thank you very much. And of course, many thanks to all consortium partners, so Institute of Human Genetics, Nęcki Institute, Poznań University of Technology, University of Białystok, University of Łódź, University of Gdańsk, Jagiellonian University in Kraków, University in Warsaw, and two national institutes of cardiology and oncology, and Łukasiewicz Port, Polish Center for Technology Development. So you can see we have really very strong consortium and the biggest universities in Poland. Yeah? Thank you very much for your contribution. And my last thanks is to our business partners. Their role is quite difficult because they cannot be beneficiaries of they cannot benefit from the project directly because we expect from them to contribute, to give some contribution, but we cannot offer them much. So I really appreciate this contribution to companies, to Polish companies, Becoders, which offers individual IT solutions for education and business, and also Analityk Genetyka, Unruk Wójtowicz, which are responsible for innovative laboratory solutions for biological and medical sciences, and their contribution will be Dragon service for data analysis. So I really appreciate and thank you very much. And that's all what I wanted to say in a short words about the project. Thank you very much again for coming here, for being with us, and I hope everything will be going on well within the next 4 years. And of course, after the coffee break, I invite you to come back here. And do we have some organizational issues? The participants of the project maybe should take their badges, yeah? They are outside in the hall, so you can find to register for the closed session of the meeting, yeah? Thank you very much, thank you to our prelegents, and see you after the break.
At noon sharp, we will start the second part of the project, so now enjoy the coffee in the hall.